Hello, and welcome everyone to this podcast on bacterial transcription. On the previous podcast, we had talked about the general nature of transcription and some of the general features of the gene and of the promoter and some of the requirements. Now I want to spend a little bit of time talking about the details associated with transcription as it relates to bacteria. And to do this, I want to first talk about promoters and then RNA polymerases. So I'm going to draw the gene sort of to how we had it before, except I'm going to focus more on this part here where the promoter is. So I'm going to draw a line here and say the ORF right here, and then the terminator is way off the whiteboard here. First I want to say that this ORF, there's this part that we're going to call the transcription, TXN for short, start site. This is where it's going to start making RNA here. It's not going to make RNA before here. And the very first base here, we always call this +1. Everything this way is downstream of the start. And we will label these bases as -1, -2, -3, -4, and all the way down. The promoter for bacteria has these two consensus sequences that are pretty important. The first one we call the -10 sequence. Not very imaginative, but that's what we call it. And then the next one is the -35, because it begins 35 bases from the start. This one begins 10 bases from the start. We call these both consensus sequences. Meaning if you look at any bacterial species, the sequence at this -10 part of the promoter and the -35 part of the promoter will either be identical or extremely similar. There will be some differences in the sequences, but not a lot. This region in between the consensus sequences are also important for transcription, but we will see many more differences between bacterial species in this region here. So if I were to ask which part of the promoter would tolerate a mutation without having a negative consequence, you might say, or you probably should say, this region here, because there's a lot of variability here. These regions here, -10 and -35, will affect transcription. While we're here, we should get in the habit of making sure we always label our ends, so 5', 3' on here, and then 3' to 5' here. We're going to be reading it 3' to 5', and we know it's going to be the bottom strand red because this is where the promoter is and this is where the open reading frame is, so we know it has to be red in this direction. Because of that, the strand that is red, we always call that the template strand because it serves as the template. The strand on top that is not red, we call this the coding strand. We'll go over later why we call that the coding strand, but for now, keep in the back of your mind that the RNA that is produced by looking at the template strand looks just like the coding strand as far as the sequence, with the exception of the RNA that's produced, instead of having thymines, which would be found in the DNA, will now have uracil. But the RNA that is produced matches the coding strand. But to produce that RNA, the enzymes have to read the template strand. Now, next I want to talk about the enzyme that binds to this promoter to allow transcription to occur. But I'm going to erase some of what we have written here so that I can draw the enzyme that binds to this promoter. In bacteria, what binds to the promoter first is going to be this enzyme that we call the core enzyme. It's a multimeric protein, meaning there are multiple subunits to it. In fact, there are going to be 5 subunits found here. We will have 2 alpha subunits, and the alpha subunits bind DNA. They are also important in providing structure to the overall enzyme. But their main claim to fame is that they are what actually binds the DNA. So let me draw these a little bit bigger. So these are the alpha subunits. The next subunits we want to talk about are the beta subunits. And they form here. And their job is that they act as the catalytic part of the enzyme. Meaning, once the alpha subunit binds to the DNA and it moves into this open reading frame, and we need to start building the RNA strand by bringing in these different ribonucleotide triphosphates, it's the catalytic subunit, this beta subunit, that facilitates that synthesis. The next part of this subunit, of this core enzyme, is called the omega subunit. And this is one of those things, even though we've been studying this a very long time, or the people who study this have been studying it a long time, there's some debate about exactly what this subunit's doing. I have pretty strong evidence that it provides stability to the structure. We also know it's not essential under all circumstances. Under just typical transcription for the bacteria, you don't need the omega subunit. Under certain circumstances, more stressful circumstances, it becomes more necessary. Now this core enzyme will bind to the DNA. And the core enzyme can bind to the DNA, and it can even move along the DNA, all's fine. However, it's not going to stop to make the RNA unless we have a sixth enzyme bind. And I just realized I didn't put the sigma subunit in. And the sigma, usually we draw it like so. That kind of holds all those together because it plays this role in stability. Well, this next enzyme that binds is called the sigma subunit. And when the sigma subunit binds, the core enzyme now becomes the hollow enzyme, H-O-L-O enzyme. So sigma will come along here and bind. And once it binds, as this as this core enzyme moves forward, it's going to stop here, but only stops at the promoter if sigma is attached to it. Sigma recognizes these consensus sequences of -35 and -10. Without sigma, the enzyme that never stops here. The RNA polymerase, the core enzyme, and then the hollow enzyme never stops. Now, a typical bacterial cell will have genes for the beta and the alpha and the omega subunits, single genes of each of them. But for the sigma subunit, there are multiple sigma subunits. So multiple genes for sigma. And the reason for that is that there needs to be some way that this enzyme knows which genes to turn on. So this adds to the regulation. Some sigma units are specific to certain genes. So this just adds to the ability of the cell to be able to regulate which genes are going to be turned on or kept off. So now I want to talk about initiation, elongation, and then termination in bacteria. So we're going to start with initiation. So let's draw our gene here. And we'll draw our promoter here. And we'll draw the core enzyme as it binds just upstream of the promoter. And remember, down here we have our ORF, our open reading frame. And as I said on the last slide, as this core enzyme, the RNA polymerase, moves over the promoter, and so now it's here, and the sigma subunit is bound to it, it will recognize that promoter and will target this gene as being a gene that needs to be turned on, expressed, and make that messenger RNA. What then happens, let me kind of blow up this region here, similar to what we see in replication. At this region here, when these enzymes bind, it will form a small bubble. However, this open region where we see these exposed single-stranded pieces of DNA, all of that is still protected by this RNA polymerase. So there's no need for these single-stranded binding proteins, because this enzyme here, this RNA polymerase, opens up the DNA, breaks those hydrogen bonds, and then, as we'll see in a moment, begins to make RNA. And as it makes RNA, this RNA polymerase is going to move. As soon as it makes that RNA and starts to move, these single strands are immediately wrapped back up into a double helix. So the single strands are never exposed. They're always protected within the enzyme. When the RNA polymerase, with this sigma subunit attached, I guess I could draw it here just to be complete. With the sigma subunit attached, it recognizes the promoter, and at that moment it binds much tighter to the DNA. And it causes what we refer to as a conformational change. Binds tighter to DNA, the enzyme changes, that's why I drew it as a square instead of a circle. This conformational change now allows this DNA to open up just a little bit and allow our NTPs to move into the enzyme and begin to make RNA. Again, in a complementary manner. So if we had on the DNA something like TAC, the RNA that's going to be produced here will eventually be an AUG. It still has to be complementary, following Chargaff's rules. And remember, over here we had 3' to 5', so this is going to be 5' to 3'. Initiation, the process of initiation, will allow between 9 to 12 base pairs form before it moves to this next stage of elongation. So this ends the initiation stage once these 9 to 12 base pairs are made. Now let's talk about elongation. Okay, now we're going to talk about elongation. And as the name implies, this is the process where the messenger RNA is going to extend and get longer. So here is our gene that we've been following for a while now. The promoter was down here. We're done with the promoter because now the RNA polymerase is moving along past the promoter. And one important part that happens during elongation is that our friend, the sigma subunit, falls off. It's not necessary for elongation. Its only purpose is to recognize that promoter. So once it's been recognized, it falls off. This allows the RNA polymerase that's left with the alpha and the beta and the omega subunits to work much more efficiently. And it does so. It's a pretty fast enzyme, not quite as fast as DNA polymerase. But the RNA polymerase, I'm just going to put RNA-P for RNA polymerase, it has a rate of about 40 nucleotides per second. I'm going to indicate some bases here along the DNA. CCT, TAC, CGA, CAA, TAC, CAT, and then ATC. During elongation, the RNA polymerase moves forward and makes an RNA that is complementary to this strand. However, it doesn't stay attached to it like we would see during DNA polymerase. So we have our RNA here. This has already come off. It is GGA. Remember, this is our 5' end. It's now forming these bonds here. So this is going to be A. The nucleotide A will come in and bind there. And then a U, because there's no T's, and then a G. And it will continue to move down this gene until it gets all the way to the end of the gene. And then the RNA that is produced, I'll just draw it like so, 5', 3', is GCA, AUG, and then here, GCU, GUU, here we have AUG, and then we have a GUA, and this last one is going to be UAG. I don't have room here, so I'm just going to write it down here, UAG. We'll come back to this later when we talk about translation. There were some important codons that I included. These 3 bases in a row we call a codon. The first one here is AUG. We'll learn later that this is the start codon. This is what's going to begin every protein sequence. And then every RNA, every protein stops making proteins at one of 3 different stop codons. And UAG is a stop codon. You'll learn that there are 3 stop codons, and this is just one of them. However, transcription doesn't stop here. The RNA polymerase is pretty good, but it's still just an enzyme. It's not very smart. it doesn't know to stop making RNA here. It's going to keep on going. There's got to be some kind of signal that tells RNA polymerase to stop making RNA. And that's what we're going to talk about next in termination. So termination. Let's draw our gene again. And we'll draw our RNA polymerase with the alpha and the beta and the omega subunits in it. And it's made this RNA. And here we have our stop codon. So we have everything we need to make a protein at this point. And over here we have our start codon. But nobody's told the RNA polymerase to stop making RNA. And so there has to be a mechanism to do that. And there are two such mechanisms. The first that we'll talk about is called row independent. The second one is called row dependent. Row is a protein, so the first one's going to not require this protein, and the second one will require the protein. In order for termination to occur, though, RNA polymerase has to stop making RNA. So let's write what has to happen. RNA polymerase has to stop. RNA polymerase has to be released. It has to fall off the DNA template. And then the RNA that's attached to this DNA has to be released. So we're going to start with row independent. I'm going to erase everything down here real quick. In row independent, what happens is as this RNA polymerase moves down the DNA, at some point the RNA that is produced will form what we call a stem loop structure. The stem here and the loop here. It's really kind of inside the RNA polymerase, or kind of engulfed in the RNA polymerase. I just can't really draw it in there to show you it. But this is a very stable structure. It contains G's on one side and C's on the other side. So the tighter the hydrogen bonds, the 3 hydrogen bonds. So it's a very stable structure. And remember, down here, we still have our stop and start. We just need to be released from this DNA template so this gene can go on and do its job. So the first step here is that it forms this hairpin loop. So let's write hairpin loop. Then by some mechanism that's not completely understood, but we know it happens, is that the RNA polymerase, here in blue, pauses. I kind of think of this stem loop as functioning as a speed bump. The RNA polymerase is cruising along, making RNA, and then all of a sudden it hits this bump, this stem loop. And that causes it to pause just a little bit. And as it pauses, what happens is, within these nucleotides right past the stem loop, the DNA has several A's. And so the RNA that's being made here would be several U's that are binding here. And so it's a long stretch of these A's, U's, which form the much weaker bonds of each having 2 hydrogen bonds. So string of A's bound to U's. This being a much weaker association and the pause caused by the stem loop is enough of an interference to make it unstable. And then it causes the RNA to be released and the RNA polymerase to be released, and transcription at this point now is stopped. So once this happens, this RNA molecule is released. Don't really have anywhere to draw it, so I'll just draw it here with our start and our stop. And now it can be translated. Now, a bit of a spoiler alert here. I'll come back to this later. In bacteria, this all occurs in the cytoplasm without a nucleus. So as soon as this start codon is made, it's already making proteins. It doesn't wait to get back into the cytoplasm. We'll cover that a little bit later when we go to translation. But just so you know in the back of your mind that E. coli bacteria in general are very efficient organisms. And they don't wait for the RNA to be completely made before they start pumping out proteins. Okay, so this is row independent. Okay, let's now talk about row dependent. It has many of the same features that we saw with row independent, in that the RNA polymerase is going to keep making RNA until it's told to stop. At some point the RNA polymerase will hit this region where it's making the RNA, and just like we saw with row independent, it does form this hairpin loop. The one difference is that it's not followed by a string of A's and U's. So even when this stem loop that's formed causes a pause by the RNA polymerase, this association between RNA and DNA isn't weak enough to have it fall off. So it would keep going. We still have our stop. Let's go ahead and be complete here. So what's going to cause it to fall off? Well, let's first label our steps here. So we have stem loop form. This stem loop causes the RNA polymerase to pause. And then one of my favorite proteins is this rho factor. It sort of acts like a cat if the RNA polymerase is a mouse. If you go with my analogy here, I'll write mouse. This rho factor binds to this region of the DNA, and let's go to name this region. This name of this region right here where it binds is called NUT. So this rho protein binds to this sequence that we call NUT. When it binds, the rho factor moves in this direction of the RNA, in a way chasing the RNA polymerase. However, it can never catch up to it. However, when this pause occurs, because of the stem loop and the RNA polymerase pauses, rho can catch up. And as rho catches up, one of the proteins in it, so rho isn't a single protein, it's this collection of proteins together that function as this rho enzyme. As it hits this region here, it acts like a buzzsaw because it has a helicase in it. And so its helicase, let me write helicase, rips through these hydrogen bonds and releases the RNA from the DNA. In that process, as rho's moving up here, it breaks these hydrogen bonds, releases the RNA, and forces the RNA polymerase to fall off, allowing this RNA to be released from this transcription unit with its start and its stop. So these are just two mechanisms that are employed by bacteria to get rid of, or I should say, to stop transcription termination. So now let's close with a quick summary. In this podcast, you should have learned about the bacterial promoter, the features of it, where those consensus sequences are. You should have learned about the bacterial RNA polymerase and the differences between the core enzyme and the hollow enzyme. You should know all the subunits involved, the alpha, the beta, the omega, and the sigma. And then we went through in pretty good detail about initiation, then elongation, and then finally termination with the row versus the row independent versus the row dependent. As you go over this material, if you have any questions at all, please let me know, and I'll be happy to help in any way I can. I'll see you in class. Bye.